As we discuss a phase 2 trial that we conducted in myelofibrosis for patients who had anemia, who may benefit from luspatercept. So this is also, as you mentioned, called the MF001 study. A lot of the luspatercept trials have this ACE moniker in front of them. But so the ACE 536 MF001 study, and here's the clinicaltrials.gov number as well. And we're going to review the study design, some of the eligibility and primary endpoints. This has been presented several times over the last couple of years. We presented the final data at the American Society of Clinical Oncology annual meeting here back earlier in the month. So the key eligibility criteria. So this study included patients with primary and secondary myelofibrosis. That includes post-PV and post-ET myelofibrosis who were anemic, whether they were receiving transfusions or not. And so you can already start to see different subpopulations within this study just by the first two inclusion criteria. Their blast had to be less than 5% in the blood, and they could not be concurrently receiving hydroxyurea, erythropoiesis-stimulating agents, IMiDs, or steroids within 16 weeks prior to enrollment. That way we could see the true effect of luspatercept on anemia in these patients. So a significant washout period of drugs we know that can improve anemia in myelofibrosis. However, patients could receive these drugs prior. So as we'll see, a lot of patients did receive prior ESA, which I think is an important point. This is the overall study design. As I mentioned, there were different cohorts of patients, and these were broken down to whether or not they were receiving ruxolitinib and whether or not they were transfusion dependent. So it makes a natural 2-by-2 table for the patients included in the study. There were, again, 4 cohorts, cohort 1, 2, 3A, and 3B. Which were complicatedly named in that manner. But nonetheless, with this trial, the goal was to really kind of look at the breadth of patients with myelofibrosis who are also anemic and to see which populations may benefit the most from addition of luspatercept to their treatment armamentarium. So each of the cohorts of patients were treated with subcutaneous luspatercept. So if they were already on ruxolitinib was continued at the same dose. They were not on ruxolitinib, they couldn't start it during the course of the study. But in addition to that therapy, luspatercept was added. And at day 169, disease response was assessed during the primary phase, again, the initial 24 weeks of treatment. And then afterwards, patients couldn't continue on for at least 3 years post-dose on this trial. Ultimately, patients who did benefit throughout the course of this trial and were still on it on the end did migrate over to a rollover protocol that included patients from a number of different studies with luspatercept. Again, the kind of description of each of these cohorts. So no red cell transfusions, that was cohort 1 and 3A. So no red cell transfusions 12 weeks prior to enrollment. Their hemoglobin levels had to be less than 9.5, recorded on 3 separate occasions within 14 days of each measurement. The other 2 cohorts, cohort 2 and 3B, were getting red cell transfusions as defined here. So again, really kind of splicing out those who are red cell transfusion independent and those who are red cell transfusion dependent. So the primary endpoint. So the primary endpoint for the study were patients not receiving red cell transfusions. So this is again cohort 1 and 3A. They had to have at least a 1.5 grams increase over their baseline over any consecutive 12-week period without a red cell transfusion. So 1.5 grams increase in their hemoglobin for 12 weeks. Patients receiving transfusions, so that's cohorts 2 and 3B, they need to have they need to be red cell transfusion free for 12 consecutive weeks. So pretty firm endpoints, pretty high bar to reach for those endpoints. Of course, there were lots of other endpoints we looked at to kind of get a full sense of the benefit of this medication, including time and duration of anemia response, frequency of red cell transfusions and transfusion dependency, symptom response, and changes in average hemoglobin. Analyses were conducted in the IIT population, consisting of all enrolled patients, whether or not they received a luspatercept. And of course, we had a keen eye on safety. Now, this drug has been through development for some time, and as Jean-Jacques mentioned, approved in other indications. So we do have a good sense of the safety signals and profile of this medication, but we wanted to keep an eye out for any unique side effects in this population.